Kia ora team. I hope you're all doing okay. Just let me know if all the sound is coming through because it's the first one of these things that I've done in a while. Our plan for today is to have a little kōrero about selective breeding and touch on some modern selective breeding methods, and then we'll talk to you about the tasks for this week, and we'll leave this video here so that you can kind of go through everything. I know the sound's not so good. I wasn't able to manage to get my microphone to work, so we're using the little baby one in my laptop, which is doing its best. But I'm sorry it's like not the best quality. As we go through this, if you've got any questions, feel free to fire them away in the chat. I'll keep flicking back to having a look at that. You're more than welcome to ask things and we'll go through that. All right. I can see we've got a bunch of us here, so let's make a start. All right. So, in terms of the work for this week, we've got some content around selective breeding that we need to understand, but your major task is to finish the first checkpoint. Now, the first checkpoint doesn't involve too much, and we'll go through exactly what that involves. We've also put up the checkpoint for next week, so you can actually get a head start on that if you'd like to, though we understand that this is a pretty challenging time for us, so take as long as you need. You've got the full 2 weeks to get those 2 checkpoints done, but we'll be checking as biology teachers after that first week to make sure everything's going okay. We're also having this live stream, and we're going to have a Google Meet later in the week. The Google Meet will just be a bit of a review opportunity, and maybe we'll play a Kahoot. So that's all on the classroom for you there. So let's have a look. What is selective breeding? Well, selective breeding, sometimes it's known as artificial selection as well. It's this method that humans have done over time to produce offspring that they desire. Not necessarily offspring that are best suited to the quote-unquote natural environment, but best suited to humans' needs. So a good example of this would be dogs. We've selectively bred dogs all over the world for different purposes, all the way from your little handbag dogs up to, big Great Danes or racing dogs or huskies. Each different dog has been bred for different purposes and reasons, and that's led to the different characteristics that they have. So a good task for you to be thinking about is just how many organisms have we selectively bred, and you don't just have to limit this to animals, but there are so many different organisms that we've selectively bred, and the reasons why are kind of the key things to be thinking about here. So for cows, sheep, pigs and goats, all organisms that we've selectively bred for farming purposes. We've bred them to be less aggressive, to produce higher yields of food, to taste better, to be able to survive in different environments. Whereas for dogs and cats, they've been bred for different purposes. So dogs, you might have bred them to assist in locomotion, to go, if it's a husky, for example, to pull a sled. If it's dogs, maybe they've been bred for racing or for aesthetics or for just companionship or for protection. So each different dog species, depending on what traits were being selected, will determine its final phenotype and what it looks like. There's a great video here for you to look at on the history of dogs, which is quite a nice little introduction to selective breeding. And that kind of brings us to talking about what we mean when we say traditional selective breeding, because that can be sort of a little bit of a dicey term. It's a bit hard to Google. But what we're meaning by that is those old school methods of selective breeding. So you can imagine if you were a farmer 10,000 years ago and you were first starting to form agricultural societies, one of the things that you would be doing is selecting the organisms that you would breed together to go on to the next generation. Now, the key things to understand here are some of our level 2 genetics content. So that's understanding that successful generations are able to pass on their genes. Their genes are then more present in the next group of the population. And over time that can lead to things like inbreeding. You can test for which genes organisms have by doing test crosses. You can produce organisms that have a different variety by taking wildly different parents and breeding them together. So those are all the sorts of things that people have been doing for, close to 10,000 years, as they've been trying to figure out how to get the organisms that best suit their environment. If you need a reminder of what a test cross is for figuring out the traits of an individual, that's taking that recessive organism, homozygous recessive, and then breeding that with the unknown phenotype to be able to figure out what ratio of offspring they've got, and therefore figuring out if the organism you've got there is purebred or not, or homozygous dominant or heterozygous, should I say. So those are sort of the things to be thinking about as we move into this. Now, we often talk about selective breeding when we introduce it with animals that we're really familiar with, but this happens with plants as well. So corn, or maize actually, would be a better way to say it, because that's the indigenous word. Maize is a plant that has been selectively bred for generations. It's a grass, but over time it was selected based on the size, the sweetness, and the ability for those organisms to be able to survive. Originally, wild corn was sort of purpley, a very dark colour, but over time, as we selected for mutations that had different traits that we desired, it lost that purple colour. Now, one term we're going to come across a bunch, and we'll talk about it briefly here, is polyploidy. Poly means many, ploidy refers to chromosomes, so many sets of chromosomes. Plants have this ability to duplicate their sets of chromosomes, and that can produce doubling up of certain genes, and that can mean that you have more of certain proteins or certain physiological processes going on in these plants, which can lead to wildly different outcomes. So you can have all sorts of different implications if you have organisms that are able to go through polyploidy. Depending on how many times that can go through, that can lead to issues with being able to produce a next generation, but often it just leads to, commonly it leads to larger organisms with more of those qualities that you're after. So a lot of our species, particularly maize, have a wide variety of these duplication events where they've undergone polyploidy multiple times, and then you have subsequent mutations in those genes. So something to be on the lookout for. You can also have this effect called backcrossing, which is where you take a plant and you cross it with its parent in order to get a reinforcement of certain characteristics. Now that can come with complications, but it can also come with benefits. You also have controlled pollination. Now this tends to be more common with organisms which are harder to pollinate. A good example of this would be saffron. So really expensive plants that are challenging to pollinate can be done manually. And in fact, for the case of saffron, still are done entirely manually. Now, for some organisms, this doesn't guarantee that you're going to get the best of the bunch, but it certainly increases your chances, which is why we undergo selective breeding. So it's a very useful way of increasing your chances of getting successful offspring. So we've got some more stuff in here in terms of other selective breeding measures. So another good example of this would be grafting, which is where you cut a section of one plant and you attach it to another plant and you end up with sort of a mixture of the two. You can also do this with tissue cultures as well. Apples are really commonly grafted in this manner, as are other stone fruits. So you can have one tree that produces a whole variety of different apples as you go in search of which ones lead to the best outcomes. Now this is a form of asexual reproduction, so obviously those organisms can't then pass on those genes to the next generation in that particular way, so you need to do that grafting process again. Now, there's a variety of reasons why we do selective breeding. That's why we've done it for thousands of years. I'm sure you can pretty much figure these ones out. But yeah, it's not just about bigger yields, it's also about disease resistance and things like that. So yeah, just as we're going through here, feel free to fire off any questions if there's anything that you're unsure about. So we talked about polyploidy just before. Some different terms you might come across in your readings refer to the number of sets of chromosomes. So normally, us normal organisms have two sets of chromosomes, half from one parent, half from the other parent. So we would describe ourselves as 2N, or diploid, di meaning 2, ploid referring to chromosomes. You can also have triploid, which is 3, or tetraploid. You can have octaploid, you can have hexaploid, and there are a variety of species that fit into those. So as you're learning about these species that you might want to write about, you might find out that your species, for example, kiwi fruit, is hexaploid. And you might be like, what does that mean? Well, that means it has 6 sets of chromosomes. So it's undergone these duplication events, these polyploidy events. Yeah, you can actually induce this and make it happen by adding certain chemical compounds which stimulate this effect. And it's a good way if you're trying to make new varieties to rapidly produce a wide variety of variation rather than just waiting for the slow tick of mutations to occur over time. So that would be an example of a modern iteration on a traditional technique, right? Using those chemicals to modernize the process of creating polyploidy organisms. You might also hear of the terms, if you're going to focus on plants, autopolyploidy and autopolyploidy and allopolyploidy. Now, auto refers to within the same species, whereas allo refers to the result of different species. An allopolyploidy species, you might take, and they give the example here of a brassica and then something a little bit different, a little bit of a different plant species, and you add them together and you get a slightly different organism. Kind of like a lion and a tiger makes a liger. So it's kind of a similar method to that, where you end up with a doubling of the chromosomes, whereas with the lion and the tiger example, you're only taking one set of chromosomes from each. Autopolyploidy is where you produce an organism within, just by duplicating the chromosomes within the same species. Now, most of these ploidy species will be infertile, particularly autopolyploidy species. So the best example of that would be bananas. You can't really create a new generation of sexually reproduced bananas. Most bananas are clones, which leads to some of the complications you would have discussed last year around where you have multiple like organisms that are really genetically similar, and so they're more prone to disease, for example. And there's a little slide here which breaks it down in case you needed some more detail on those. There's also this great video which talks about selective breeding, another example in dogs, which is really good. But there are a few other modern techniques that are probably worth discussing. Now, this particular technique here is referring to artificial insemination. So artificial insemination is where sperm are delivered by humans rather than by the organisms themselves, and that can be done in a variety of different ways. And you might want to do that if you say the bull that you wanted to breed with your cows lives on the other side of the world. You might buy from like a catalogue online. But also a more recent technique, which I think is really, really neat, was developed actually at the University of Auckland by Professor Cather Simpson, and the work of her team and the work of Engender Technologies, which now is, you know, a multi-million-dollar company. It's going to be real big business, this stuff. They sort the sperm into male and female using lasers. Specifically into X and Y would be more scientifically accurate. So splitting into X and Y, and what that means is that you can get a vial of just X chromosome sperm, meaning you don't produce bobby calves. Now, if you produce bobby calves, what do you got to do with them? You got to send them to the freezing works, right? You can't really use them if you're a dairy farmer to produce milk. So it's really beneficial if you can guarantee that success. Take that, even if you're taking that 50-50 chance down to a 80-20 chance. That's still, you know, millions of dollars a year that, you know, this could be saving the New Zealand economy and dairy farmers around the world. So this is a huge technology that's coming up in the not too distant future, that farmers around the place will be using these sperm sorting technologies to be able to produce consistently only those dairy cows rather than bulls, for example. But this could be done for any species. Now, there's some interesting like ethical things to consider. we probably wouldn't want this happening in people, but that's kind of this is something we don't really talk about too much, but we still want you thinking about with regards to this internal. So it's really, really cool, this technology, though. So if you want to learn more about it, there's a link there for you to look at. And there's also a video in the description about the processes of artificial insemination. There's also some more detail here, which talks about that process, and that comes from the ESA book. The whole chapters of those are available for you if you want to look at them. Okay, so there's some more detail here about how exactly sperm are stored for your interest, or for those of you who are interested in writing about this as a modern interpretation of selective breeding. So once you've talked about what selective breeding is, you could then talk about this if it applied to your organism that you chose. Now, a couple more modern techniques to talk about. Genomic selection. So this is a really good one around how do you make sure that the organism you're selecting to pass on to the next generation is the best. So for this one, what you do is you get your, usually you have your own farm, and you would go to a scientist who's already done this work. And what the scientist has done, or what the previous people have done, is they've got a reference population. They've looked at the quality of basically everything in these organisms. So they'll look at how good the meat is, how quickly they grow, how much they put on muscle, all of these sorts of things. And then they've gone through the genotype, and they've genomically sequenced, or at least looked for genomic markers within each of these organisms. They've then made up a prediction model, which says that we think an organism that contains all of these markers on their genetic code is likely to be a really successful organism with regards to whatever the farmer's after. If the farmer's after a pig that can put on weight really quickly, or can grow really, really large, or has a particular kind of taste profile that they're after, then what they do is look for certain genetic markers. And you, as the farmer, can refer to that prediction model and then genomically test all of your own pigs, and then figure out which ones are most likely to lead to superior outcomes for the next generation. So that genomic selection allows you to make a more informed choice than just looking at the surface level characteristics. You're actually looking at the genes and markers within the genes. So that is an example of how this would be beneficial. Now, you don't just have to do this in pigs, this happens in plants all the time as well. So this is a good one to be looking at if you're looking for a more advanced method. And there's a few terms you might come across, so it's worth us talking about here. So you can use artificial intelligence to make those selections more accurate. You can also talk about the particular genetic markers, so those could be genes themselves, so looking for a particular version of a gene, a particular allele, bringing it back to that level 2 stuff. You can also look at these things called quantitative trait loci, or locus if it's one, right? Which is just a spot on the DNA sequence. And usually it'll be a base or a couple of bases that are flipped in some way. And sometimes they'll have a direct reflection to some sort of outcome. So they might be on a gene that's correlated with muscle growth, for example, but they might not be. They might just be things that tend to get selected along with these other traits. Now, the point is, we don't know. We just know that these particular spots tend to correlate with organisms that have desirable characteristics or undesirable characteristics. So those spots, those quantitative trait loci, can be what we call single nucleotide polymorphisms. Single nucleotide, one nucleotide, right? Polymorphism, meaning changed, right? So it'll be an A instead of a T, or it'll be a C instead of a G. And so if you find the right SNPs, SNPs, in the right places, that might tell you that the organism that you're looking at is desirable. There's also another term you might come across called microsatellites, which is when you get these kind of repeating short bits, and the length of those repeating short bits, or how many you have, or where they're located, can sort of come across with different genes from different organisms. Normally, microsatellites are, non-functional bits of DNA, but if they're next door to, say, a desirable gene, then maybe you've got this big obvious microsatellite that's being carried across with your genes, so you can just go looking for these microsatellites that are markers of a good next generation. So each of those different methods, be they SNPs or be they just other forms of genomic selection, could be another modern method for you to talk about with regards to your organism. Now, to understand why we talk about SNPs, now 90% of our DNA isn't coding directly for proteins. There's a variety of reasons why we think it's like this, but the long and the short of it is that we can still look for those differences between one organism and another and look for correlations. So maybe you find that organisms with this particular single nucleotide polymorphism, this particular SNP, tend to be larger, or tend to be more muscly, or tend to put on weight more quickly. And so therefore, you would look for an organism that has that SNP when you're selecting your organism for the next generation. Hopefully that makes sense. There's a lot more detail to SNPs, and there's a lot of geneticists who spend their whole lives looking at these things and developing mechanisms for detection. You don't really need to know about them, but they're really cool. You would want to talk about some of the specifics, and if you're going for merit or excellence, you're talking about why do they do what they do, right? And we'll come to that in a moment when we talk through the checkpoints. So there's some more details here and in the ESA book about detecting SNPs or detecting those differences between organisms. So yeah. The last sort of one I thought we should talk about is embryo selection. So this is another modern technique for getting organisms that are desirable in the next generation or selective breeding. So imagine you've got an organism, say a cow, and you've got a bunch of embryos, you've taken them out. What you can do is look at them. Either look at them morphologically under a microscope, or you can look for evidence of polyploidy, you can test them in some way, or you can use genome sequencing to figure out which one you should go for. And that can be really beneficial, particularly in organisms that it costs a lot of money or it takes a lot of time to breed. So yeah, essentially what it involves is you remove the egg, usually you can remove the whole embryo actually from that organism, and then you screen it to look at which embryo you're going to select, and then you can add it back either to that original organism or to a different organism. Yeah, so they describe that process there, and there's a variety of reasons why you would do that, but essentially it's just another way of selectively breeding organisms that are desirable. Now, this particular method can have quite high failure rates because it is an invasive process involving removing those embryos and putting them back in, but it might be better off to do that if you get a way greater production, even at the expense of that cost, right? So there's swings and roundabouts to these things. So with regards to what you have to be doing with all this information, your job for this week is to select your organism, and there's some stuff on the classroom to help you out with picking an organism that would suit you. When you've done that, your job is to describe selective breeding, so whatever your definition of it is, and talk about what it means to do traditional selective breeding in your organism. Now, what we mean by that is that that's that historical stuff that we were talking about initially, and then once you've done that, you want to describe one method used for selective breeding using some biological ideas. So that would be any number of those methods that we just talked about, but you'd want to pick one that's related to your organism where you can find references that suit it. So you might talk about the genomic markers use in pigs. You might talk about polyploidy selection in plants. You might talk about embryo selection in cattle. So any of those would be a valuable thing for you to write about. Now next week, we're going to be working on the stuff that's in blue here. So the stuff that's in blue involves talking about these more modern methods. So, I mean, you can get started on that if you'd like to, but you can dive into more detail on that next week. And you also want to be explaining why those ones are done as part of your sort of merit to excellence level explanation. So, how does this benefit the farmer? How does this benefit the selective breeder? How does this affect the organism? And then the most important part of this is the two biological implications. So that might be implications to survival, it might be implications to the survivability of the species or health complications or all that sort of stuff. But that's what we're going to talk about now, okay? So hopefully we remember from our previous kōrero about this, that there are a large group of different biological implications that we might talk about. And by the way, as we're going through this, welcome to fire off any more questions in the chat. I'll keep flicking over from time to time and checking it out. But there are 5 major types of biological implications that we like to talk about, and this is what you're working on for that second week. So we've got ecosystem, so impact on the ecosystem, impact on genetic biodiversity, so that's our genes, but we'll go through each of these in sequence. The healthful survival of individuals, so that one's quite an easy one to see for some of these ones. How different populations are able to survive or not survive, and how this affects their evolution over time. So, an example for health and survival of individuals would be our Belgian blue. Now, the Belgian blue are these super muscular, super lean looking beef cattle. They are able to pack on the muscle really quickly due to this particular genetic mutation, which if you're interested, you can give some research. It's called double muscling. And those with the double muscling gene are able to pack on this muscle really quickly, which makes them really desirable. They're also very lean, so they produce a very particular cut of meat, which can be really useful at market. The problems start to occur when you think about trying to give birth to one of these organisms, right? So a large proportion of calves that are born that are these Belgian blues need to be born by C-section, which requires having a vet on site to be able to do this. There are also some other health complications, but the C-sections is the main issue there, and that's due to the small size of the birthing canals relative to the size of the organisms. So obviously, if you selectively breed Belgian blues, there are implications biologically to the health and survival of those individuals. Hopefully that makes sense, yeah? There are also implications to biodiversity, and this is something you might have touched on last year when you were looking at things like bananas. If you develop a monocultural, so like you only have this one very specific type of organism, that makes them more vulnerable to ecological collapse, which is when disease hits, and then all of a sudden you can't grow that variety anymore because they'll just get the disease and die. The Irish potato famine comes to mind. Yeah, so we'll go into the other ones at a later stage, but if you've got any questions around them, you're more than welcome to ask. What we've got on the next slides, and I won't go through this, but I'll leave them there for you to look at, is an annotated exemplar going through one of these examples, and you'll see different sections of this are highlighted where we're pointing out that they've met those different requirements. And you'll notice there's a bunch of writing to do here. So this person's talked about marker-assisted selection, but it's not a huge amount, and that's it. So 1, 2, 3, 4 paragraphs to write, and they've sort of tackled different parts in each. Yeah, so we've given you also a few different examples of biological implications for selective breeding, just to get you thinking and to help you to get started with this. So I'll let you look at those in your own time. We've also given you a bunch of resources here, and Miss McNair, Miss Misa and I have been trying to put together some resources which we will give to you to help with that second part of the checkpoint. All that I ask is that you take a look at this, which are the success criteria when you're finishing off this internal. So you'll notice that by the end of this internal, we will have talked about one manipulation or one example of selective breeding, one example of transgenesis, 2 biological implications of selective breeding, and 2 biological implications for transgenesis, and that's your achieved. So there's not a huge amount to write there to get the pass. To get the merit, it's about talking about why we've done it. It's also about talking about biological implications in an explained sort of way, some more detail. Same with transgenesis. But then for excellence, it's about talking about why we've done this using biological ideas throughout, and then discussing what the implications are when you are comparing your selective breeding and transgenesis. So it's about comparing and contrasting, right? But that's basically it for today. I know that's a lot of content, but I'm hoping that this will also serve us for next week as well, because this basically covers all you need to know, pretty much, for this internal, at least for starting it around selective breeding, and we'll go through the transgenesis stuff at a later stage. Lastly, later this week, we'll be having a Google Meet where we'll just do a bit of a Kahoot, happy to answer any questions, and we can go over these things. If you have any questions relating to this, you can leave them in the chat or in the comments. But other than that, have a lovely rest of your week. Look after yourselves and your whānau. Keep wearing a mask and social distancing and all of those sorts of things. If you need anything from me, please don't hesitate to let me know. Okay? All right. Have a lovely rest of your week. Look after yourselves, and I'm sure we will chat relatively soon. Okay. Ka kite team.